1 in 5 people will at some point in their lifetime experience a sort of clinical depression. I believe that what we're learning from studying drugs like ketamine and psilocybin is that they provide researchers like me an insight into mechanisms that mean we can make better treatments. My name is Emma Robinson and I'm a Professor of Psychopharmacology at the University of Bristol. My research is particularly interested in emotional behaviour, so I'm interested in both the basic mechanisms that underlie how the brain processes emotional information and how emotions influence behaviour, but also then in an applied way looking at the field of depression. For everybody it is slightly different. In fact, that's one of the challenges we face in the field, is how do we define something that is subjectively experienced by a person. But when you look at the way it's diagnosed, it's based on things like loss of interest in rewarding activities. So things that previously people would have enjoyed, they don't get the same feelings of pleasure, enjoyment from. And also their mood is low, so they're thinking about negative things more. They feel sad and they don't necessarily know why. We treat depression really in 2 ways. We have psychological type of therapies, behavioural therapies. The other thing we have is drug treatments, pharmacological treatments, and these are the serotonin specific reuptake inhibitors, but the problem with them is side effects. Also, they will take several weeks to work. And that's not great when people have a mood disorder and are feeling very sad. You want them to be treated quickly. Ketamine, that, if given to patients, works really quickly. Within 24 hours, people start to feel better, and it lasts for several days. And we also have the clinical trial going on with the psychedelic psilocybin, which will deliver its outcomes in the next 12 to 24 months. And then we'll see whether we have another drug that can potentially change mood very quickly and also have long-lasting effects. So ketamine is a very different type of drug. It's traditionally been used as an anaesthetic. Very, very useful drug. It works on part of what we call the excitatory transmitter system of the brain. Glutamate is the transmitter. It works by modulating. So it seems from our data that at low doses ketamine is blocking this receptor, but only really having effects on some sort of more sensitive parts of the brain. And those seem to be the emotional regulation parts of the brain. That's what we think is happening. So there's lots of psychedelics that come from plants. This is one that's found in mushrooms, so it's commonly associated with magic mushroom. And when it's ingested, so when people eat it, consume it, it's turned into a substance called psilocin, and that is the thing that has effects in the brain. It works on the serotonin system, but a bit different from the antidepressant drugs that we were talking about, those serotonin-specific reuptake inhibitors. What it does is it interacts with the receptor, and our data's pointed towards there being an important role for a number of these serotonin receptors in how psilocybin or psilocin is acting in the brain. We do some of our work using slices of brain to study the electrical properties and how neurons are behaving. But we're interested in behaviour, and it's very hard to model that using a cell system when you're wanting to ask that really complicated question. How does this part of the brain affect emotional behaviour?
All of our animals are kept in red light because the majority of our work is behavioural testing. and we want to be doing our tests in the animal's active phase, so we're not waking them up in the middle of a sleep cycle to ask them to do a complicated task.
Certainly the rat and mice have the right bits of brain and the circuits that we know are involved in regulating those behaviours. We're interested in something quite complicated, which we call affective biases. That's been the critical thing we study. That happens in humans. We all experience it. You have a bad day, you just think about things more negatively. It's that glass half empty, glass half full idea. We ask our animals to learn 2 bowl digging tasks. So we have rats and they go down to 2 bowls with 2 different digging substrates. One's got food in it, one doesn't. And they have to learn which substrate predicts finding the food. We change the position of the bowls. One of those tasks is manipulated with some sort of stressor or something positive like social play, and that creates what we call an affective bias. A good day for a rat would be a day in a playpen with its mates. We know that if you put 8 rats in a big pen with toys and objects and friends, that would give a positive affective bias. And antidepressants look very similar. So if you give an acute treatment of an antidepressant, you see something really similar. A bad day for a rat would be 10 minutes in a small box. It's stressful for them and it generates a negative emotional state that then we read as a negative affective bias. So they generate these 2 independent memories, and then the readout is 30 trials where they get presented with the 2 digging substrates they've experienced before. One was under a affective state manipulation, one's the control, and we just count what their choices are. We keep the value of the experience identical, so they should like both. But we found the digging substrate encountered when they had a negative experience, they will actually have a relative reduction in how much they choose that, and vice versa for positive. So we see positive and negative biases. That is the model we work with because we know that in humans, negative biases is a pervasive neuropsychological impairment that is seen in people with depression. What we then can do is we can then ask the question, how do antidepressants change this behaviour? So we know that conventional antidepressants, these delayed onset antidepressants, they work by changing that bias. So they make things more positive, but they only make things more positive that are newly learnt. They don't have any impact on past experiences. The very surprising in some ways, but exciting thing is that what ketamine we found was able to do was to change those past experiences. So where you had a negatively biased memory, we could give ketamine before we asked the animals which do you prefer, and they no longer had this negative bias. And we've been able to kind of resolve this. So it's not changing memory. The animals are not remembering differently, but what they are doing is they are not having that same emotional component to that memory. And so that's why we think it may be that these drugs are having some selective effects on circuits that generate affective biases. So what we do is we can combine our behaviour with techniques that allow us to image the brain or to record information from the brain. One of the techniques that we've set up recently is a technique called in vivo amperometry. This is a technique where we put small probes made out of carbon paste and they go into the brain, into different regions, and they can listen to the oxygen signal. Now that's the same sort of signal you're getting in an fMRI scan in a human. So we plug them in so we can record that oxygen signal, and then we can present them with a task, either the bowl digging task or a task on a screen where we have touch screens. So the rats can actually touch symbols and follow rules, and we can look at how the brain's responding in the real time to those pieces of information or to the animal's behaviour. Now we know these things, we've now got to the stage where we're asking, what could we be targeting to make the next antidepressant that's going to work quickly, have minimal side effects, be useful for a wide population? I hope we will also, though, recognise that there is a rapidly increasing problem in our society with poor mental health, particularly emotional disorders, depression and anxiety. It will always be important to have drug treatments, and the better we can get them, the better for those people who become unwell. But ultimately, our goal should be to stop people getting unwell and understanding those mechanisms that drive it. And I think that people don't think animals are useful for that. I think that's the complete opposite, because actually we can ask those detailed questions about how the brain is working and how it changes in, say, a disease model, or how a drug treatment might interact with those mechanisms.